In this lecture, we will discuss the metabolism of lipids and fats and the other metabolic processes which generated the energy utilized by the microorganisms. For most of the microorganisms, the glucose is the major source of energy, and most of the energy generated by the metabolism of glucose. But lipids and proteins are also useful resources to generate energy, and microorganism utilizes the fats and the proteins to generate different form of energies and utilize in the function of the cells. The metabolism of fats are based on the two phases. First one is the catabolism of the fat and then the beta oxidation. A fat molecule is composed of glycerol and the fatty acids, and glycerol and fatty acid combinedly represented a fat molecule. In metabolism of fat, there is a hydrolysis of fat molecule by the addition of water molecule resulted splitting of glycerol and the fatty acids. The glycerol is metabolized by the glycolysis, and the fatty acid which are in even number of carbon atom, that is 14, 16, 18 and 20, undergo the metabolic pathway known as the beta oxidation, in which two carbon pairs are removed by the reaction. This is the catabolism of fat, in which hydrolysis resulted the splitting of glycerol and the fatty acid. As you see, a fat molecule by the addition of water molecule or the hydrolysis in the presence of enzyme split it in glycerol molecule and the fatty acid molecule. This fat molecule is splitted this CH2O, CHO, CH2O by the addition of hydrogens here and resulted a glycerol molecule, whereas the hydroxyl groups are attached to form a fatty acid molecule, and this overall catabolism is the breakdown of the fats. Laterly, the beta oxidation is begun by the combining of coenzyme A to the fatty acid. Once the fatty acid is combined with the coenzyme A, oxidation of the beta carbon, that is the second carbon from the carboxylic group of the fatty acid, resulted a release of a molecule, that is acetyl-CoA, and formation of fatty acid shorter by 2 carbon atoms. What actually happened in beta oxidation is a 2 carbon splitted from the fatty acid chain and combined with the coenzyme A results a formation of acetyl-CoA. It means acetyl-CoA is comprises on 2 carbon atom molecule, and resulting fatty acid is shorter by 2 carbon atoms that are attached in the acetyl-CoA. The process is then repeated and another acetyl-CoA molecule is released. The newly formed acetyl-CoA is then oxidized by the Krebs cycle to obtain additional energy. So the process again repeated and 2 carbon atoms detached and another acetyl-CoA is generated by the addition of coenzyme A. This is the all process of fat metabolism, in which this fat molecule, which contain the glycerol and the fatty acids, after hydrolysis by the addition of water molecule is split into glycerol and fatty acids. This glycerol molecule is undergo glycolysis and produces the energy, and this fatty acid, as you count it, it is 14 carbon fatty acid combined with the coenzyme A. This is the combining fatty acid with the coenzyme A, and again another coenzyme A is adding here, and this is the beta oxidation has occurred, and these 2 carbon are detached from the fatty acid and represented a molecule of acetyl-CoA. Again the beta oxidation is repeated and further 14 carbon fatty acid resulting a 12 carbon fatty acid and then 10 carbon fatty acid. And these acetyl CoA will further involve into the Krebs cycle and produces the additional energy. So this is the whole metabolism of a fat, in which a fat molecule is first hydrolyzed into glycerol and the fatty acid. The glycerol undergo the glycolysis and the fatty acid by the addition of coenzyme A and the beta oxidation process splitted two carbon that combined with the coenzyme A resulted a molecule of acetyl CoA. As you see, the acetyl CoA molecule represented two carbon and a coenzyme A. In protein metabolism, it is also a catabolic process in which protein is first hydrolyzed into amino acids and the peptides by the proteolytic enzyme. These proteolytic enzyme actually splitted the proteins. Amino acids are further deaminated. It means the amino groups are removed, and these deaminated molecules are entered into the glycolysis, fermentation and the Krebs cycle. This is the protein catabolism. In this, a protein or the peptide molecule after the hydrolysis means the addition of water molecule converted into peptide molecule and amino acids. These amino acids are undergo the deamination. It means the amino group will be removed, resulted by the reduced NAD. This is the energy resource, and this split it into the ammonia plus the organic acid. These organic acid are two carbon organic compound which undergo the glycolysis or the citric acid or the Krebs cycle. This ammonia is toxic for the cell, so it should be converted into the urea or the waste product. So ammonia by the hydrogenation converted into the ammonium and then further into urea. So this is all the protein catabolism. This scheme representing a combined effect of all energy resources and how the energy process continues by utilizing protein, fats and the carbohydrates. Proteins in the form of amino acids, fat in the form of fatty acid and glucose in the form is the product of carbohydrate. These all glucose undergo glycolysis, protein catabolism, fatty acid oxidation, and the resultant is the acetyl CoA. This acetyl CoAs undergo the citric acid cycle in the oxidative phosphorylation and undergo the electron transport chain. Energy resources has been created in the form of ATP, which utilized by the bacterial cell for the further processes. This is also a schematic representation of the food or the resources in the form of fats, carbohydrates and the proteins. Fats are split first into the fatty acid and the glycerol, the carbohydrate, the glucose and the other sugars, and the protein into the amino acids. Fatty acid undergo beta oxidation, glycerol undergo the glycolysis, glycolysis resulted the pyruvate, pyruvate is then converted into the acetyl CoA, which then undergo the Krebs cycle. These amino acids are also converted into the pyruvates or the acetyl CoA or directly incorporated into the Krebs cycle, and this beta oxidation also resulted the formation of acetyl CoA as we previously discussed. The Krebs cycle liberate the carbon dioxide and electron undergo to the electron transport chain and reduces this oxygen into the water and generated the ATP. And these ATP, which we have discussed in class, that an electron transport chain generated 34 ATPs. In one electron transport chain, 34 ATPs has generated. The other metabolic processes actually is the utilization of light or the chemical source. Included photoautotrophy, photoheterotrophy, chemoautotrophy. What is the photoautotrophy? Photoautotrophy is the process of photosynthesis. It means in the presence or the stimulus of light, a bacteria is able to produce its energy source. There are two types of photoautotrophy. First one is the light dependent reaction and second one is the light independent reaction. In light dependent reaction, first categorization is the cyclic photophosphorylation and second one is the non-cyclic photoreduction, that is the photolysis. Light independent reactions are also known as dark reaction. What happened in the photosynthesis? Photosynthesis required a basic molecule, that is the chlorophyll. This chlorophyll generated the energy in the presence of light. So what happened in the cyclic photophosphorylation? It means a process is in continuation. When the term cyclic is used, the process is in continuation. What is the continuation? The whole process is not stopped. This is in continuation, so it is known as the cyclic photophosphorylation. Why the photophosphorylation? Because it happens in the presence of light, means photo, and phosphorylation means the addition of the phosphate group to produce the energy in the form of ATP. So in start, a stimulus of light initiated this chlorophyll and excite the electrons. These excited electrons are undergo the electron transport chain. The electron transport chain is the continuous moving of the electron to the various cofactors and the coenzymes. This is the path of electron continuously. The electron is passes. This whole arrowing is the path of electron, and these molecules are the electron carriers. It means they carry electron. These excited electron come to this molecule or the carrier, then it passes to the next carrier, it passes to the next, continuously it passes and return back to the chlorophyll. During this all process, an ATP is generated, which is the form of energy by the addition of inorganic phosphate to the ADP. This is the non-cyclic photophosphorylation. It means there is a stimulus of the light, there is a phosphorylation, but it is not a cyclic process. It means the process started and end, but not in continuation. So a chlorophyll excited electrons in the presence of light, and these excited electrons following the path of electrons towards the carriers, and these carriers produces the energy in the form of ATP and also form a molecule NADPH. This is the reduced form which is able to oxidize and provide the energy. This is the photolysis of the water, which break down into electrons, which came back to the chlorophyll, and half of the oxygen molecule and 2 hydrogen molecule, which came here to reduce this NADPH. So the energy is generated in cyclic and non-cyclic photophosphorylation. This is the combination of light and dark reaction. What is the difference between the light reaction and the dark reaction is very simple. The light reactions are happen in the presence of sunlight, whereas the dark reaction happen without the presence of sunlight. And in darkness, these reactions capable to produce energy for the bacteria. So this is the relation between the light and the dark reaction. The light reaction, which we discussed previously, produces the NADPH, which I explained, a reduced form able to provide energy in the form of electrons or the hydrogen, and this ATP can be utilized and further provided the energy. So what happened? The products, for example the ATP or NADPH, which are produced by the light reaction, can be utilized by the dark reaction and reduces this carbon dioxide by using this NADPH, and the ATP reduces it in the form of carbohydrates. So the products of light reactions utilized by the dark reaction and reduces the carbon dioxide in the form of carbohydrates or the glucose. So this is all about the metabolism of fats and proteins and the other metabolic processes, especially the photoautotrophy, in which we discuss the light reaction and the dark reaction and how energy is generated by the electron transport chain and transfer further to the dark reaction for the generation or the product of carbohydrate. These carbohydrates are undergo glycolysis and converted into energy resources. So if you have any query regarding this lecture, you can ask on the comment section. Thank you so much.